So in this lecture, we'll be talking about the cytoskeleton and cell movement. So yesterday we talked about the actin microfilaments. Today we'll talk about the microtubules and intermediate filaments. So microtubules are the second predominant component of the cytoskeleton following the actin microfilaments. They are rigid hollow rods. So they really look like pipes. They're empty from the inside. They are quite dynamic structures, meaning that they continually undergo polymerization, depolymerization, or growth and shrinkage at like what we will see in a few minutes. They have several functions. They are important for maintaining cell shape. They are important for cell movement, but mainly, really, they are involved in transporting and stabilizing the positioning of organelles. Particularly, we'll be talking about movement of vesicles, and they are important during cell division and separation of chromosomes. So microtubules look again like long rod, hollow rods. So they look like a pipe, and they are empty from the inside, like I said. Now they are made of two proteins, alpha tubulin and beta tubulin. Now, and they sort of like alternately organized. So you have alpha, beta, and so on. There's a third tubulin protein known as gamma tubulin, and it's mainly localized in centrosome, or the microtubule organizing centers in cells. Okay. So now what happens is that you have the formation of these dimers, the alpha and beta, and both of these proteins, alpha and beta, are GTP bound. Now what happens is that these GTP bound dimers can form the larger structure of the microtubules. Once they form the microtubules and once they associate with a large structure, once they polymerize, GTP of the beta tubulin only is hydrolyzed, becoming GTP bound, and GTP bound beta tubulin are less stable, so they tend to dissociate from the larger microtubule structure from the other end. So just like actin microfilaments, microtubules are also polarized structures, meaning that they have a plus end and a minus end. The plus end where you have growth and polymerization, and the minus end is where you have depolymerization. Okay, so once they are released, they undergo nucleotide exchange or GTP exchange. So GTP dissociates and GTP is bound to alpha, to mainly beta tubulin. Okay. So they undergo continuously, like I said, treadmilling. Okay, so treadmilling is basically when you have polymerization on one end, the plus end, and depolymerization or dissociation from the other end, or the minus end. Okay, so remember that it's the GTP that is bound to beta tubulin that is hydrolyzed to GTP, weakening the affinity, the interaction between the different dimers, and you have dissociation or depolymerization. So there's what is known as dynamic instability, meaning that there is a growth and shrinkage of microtubules all the time. So microtubules undergo growth when the rate of polymerization is faster than the rate of depolymerization. On the other hand, they undergo shrinkage when the rate of depolymerization is faster than the rate of polymerization. This is particularly true when depolymerization starts at the plus end, and this is called catastrophe. Okay, now our understanding of microtubules and how they are formed and how they are degraded has allowed us to come up with drugs that work on microtubules, and these drugs can either be used experimentally or therapeutically. So we have colchicine and colcemid, for example. Both of these bind tubulin, they inhibit polymerization, and they block mitosis, and these two chemicals are used experimentally in laboratories. Now similarly, we have two drugs known as vinblastine and vincristine, and both of these do the exact same thing. They bind tubulin and they prevent polymerization, and both of these are used in the treatment of cancer. Also used in the treatment of cancer is Taxol. But what Taxol does is that it binds to microtubules and it stabilizes microtubules so they do not undergo depolymerization. Now, depolymerization is important during cell division. So yes, we do need microtubule formation so that we can have the formation of the mitotic spindle network in cells, in dividing cells. But at the same time, they need to depolymerization they need to be depolymerized so that cells can undergo, they can finish and complete cell division. Now, this process of dynamic instability, polymerization, depolymerization, is regulated by proteins collectively known as microtubule associated proteins. Among these proteins or MAPs, you have polymerases, and polymerases can regulate growth and shrinkage at the plus end, so they allow for growth of microtubules. On the other hand, you have depolymerases, and what they do is that they act on the plus end, inducing or stimulating shrinkage or catastrophe. Now, once you have catastrophe going on, you have a third protein known as clasp, and what clasp does is that it binds to microtubules and it blocks and stops catastrophe or shrinkage of microtubules, allowing for the regrowth of microtubules. So what they do, what these proteins do is that they rescue microtubules. Now, there is a structure in cells known as centrosomes, and centrosomes are microtubule organizing centers. So you have this center right here near the nucleus in a non-dividing cell, and from here you have extension of microtubules. So you have the minus end of microtubules at in centrosomes, and the plus end extending outward. Now, during mitosis, centrosomes separate, or they duplicate. They become two organizing centers, extending microtubules and forming the mitotic spindle. So right here you can see a cell with a centrosome right in the middle, and extending out of centrosomes you can see the microtubules in green. Now, in neurons, things are a little different, because what you have in neurons is that you have dendrites and you have axons. So there are these two processes that have different functions. Now, what happens in dendrites, for example, is that they receive a signal from a cell, neighboring cell. Axons transmit a signal. Now, looking at the organization of microtubules, they are produced from centrosomes, but then they are released. In dendrites, they are oriented in opposite directions. So you have filaments that where you have the minus end in the cytosol and the plus end extending inside the dendrites. And you also have filaments going the opposite direction. So you have the plus end in the cytosol and the minus end in dendrites. Okay. On the other hand, in axons, you have microtubules extending in one direction only, where you have the minus end in the cytosol and the plus end always going towards the axons. Okay. Now, what's interesting is that it seems that these are organized by proteins, by specific proteins. So, for example, in axons, the microtubules in axons are associated with a protein known as tau. And this protein, by the way, is really important clinically, because it's one of those proteins that are degraded or misfolded and aggregate in the brain, causing Alzheimer's, or they are associated with Alzheimer's disease. Now, microtubules are associated with specialized proteins known as motor proteins. Just like myosin in actin, which is responsible for contraction of the actin filaments, you have motor proteins also associated with microtubules. And two of these proteins are known as kinesin and dynein, and they move in opposite direction. Now, both of these proteins are composed of a head and a tail. The head is associated with the microtubule itself. The tail, on the other hand, can interact with vesicles, organelles, macromolecules, and so on. And they are responsible for transporting and localizing and positioning these organelles or vesicles or macromolecules. So as I said, they move in opposite direction. So you have, for example, dynein, for example, it moves towards the minus end. On the other hand, kinesin moves towards the plus end. So, and that makes sense if you consider axons, for example, where you have microtubules going from the minus end inside a cell to the positive end inside axons, all the way to that terminal, to nerve terminal. So axons, so kinesins can interact with vesicles that contain the neurotransmitters or the enzymes responsible for synthesizing neurotransmitters. These vesicles move via kinesin from the cytosol, from the cell body, to the nerve terminal. Okay. Now, microtubules are also responsible for positioning organelles, specifically also the motor proteins, kinesins and dyneins. So both of these proteins can transport selective mRNA molecules in cell. So, for example, you don't have synthesis of proteins or translation going on randomly anywhere in the cell. Rather, you have translation happening at certain positions in the cell, and particularly where this protein that is synthesized is needed, whether it's needed inside the cell or out or in the periphery of the cell. Kinesin is responsible also for organizing the position of endoplasmic reticulum, lysosomes, and mitochondria. Dynein is important for positioning the Golgi apparatus in the center of the cell. Now, there are diseases related to the defective kinesins. An example is amyotrophic lateral sclerosis. Another is Charcot-Marie-Tooth disease. Okay, so remember we talked about CMT disease previously in the nucleus lecture, and we said that it can be related to mutations in the lamin A protein. Okay. Now, something interesting about vesicular movement in microtubules is that microtubules are necessary for long range movement of vesicles. But these microtubules do not reach the plasma membrane. Rather, what you have in here in the plasma membrane is the actin microfilaments. So what happens is that once these vesicles reach the end of microtubules, they change horses. That is, they change filaments. They move from the microtubules to the filaments, and that's how they can reach the plasma membrane, fuse with the plasma membrane, and release their content. Okay? So let's talk about intermediate filaments now. Now, intermediate filaments have a diameter that is intermediate between actin filaments and microtubules, and that's why they are given this name. Now, the main function of intermediate filaments is to provide mechanical strength to cells and tissues. So they maintain the integrity of cells and tissues. Now, these intermediate filaments can be divided into 5 groups. Now, these 5 groups are known as types 1 and 2, type 3, type 4, and type 5. Type 1 intermediate filaments are acidic proteins. Type 2 intermediate filaments are neutral or basic keratins. Now, in tissues, you can have a combination of types 1 and 2 intermediate filaments in mixing with each other or integrated with each other. Now, these keratins, by the way, both types 1 and 2, can form hard keratins or soft keratins. Hard keratins like what you see in hair, nails, and horns. And soft keratins are present inside cells, epithelial cells. Now, remember also that hard keratins are hard as a result of the formation of disulfide bonds. So the more disulfide bonds exist in intermediate filaments, the tougher the protein. Now, type 3 intermediate filaments include vimentin and desmin. Vimentin is present in fibroblasts, smooth muscle cells, and white blood cells. Desmin is specifically found in muscle cells. So one phenomenon that exists in cancer is what is known as epithelial mesenchymal transition. During the progression of cells into cancer cells is that they become more mesenchymal or more fibroblast-like. So they change characteristics and features from epithelial to mesenchymal, and one of the things that happens is an overexpression of vimentin. So they become mesenchymal-like cells. So type 4 intermediate filaments include neurofilaments, and these are found in neurons. Let me repeat this. So type 4 neurofilaments are found in neurons. Type 5 intermediate filaments are the nuclear lamins that make up the nuclear envelope, and we already talked about that. So these are the different types of intermediate filaments. Now, only focus on the ones that I've mentioned, okay? Again, another look at intermediate filaments, their types, their localization in different cells. So, how about the structure of intermediate filaments? How do they look like? They have an alpha helical rod structure, okay, long structure, and they have an N terminus and a carboxy terminus, head and tail. And they have a certain specialized structures, as you can see in here. So these are really domains. The way intermediate filaments are formed is that you have a chain, and then it forms a dimer. And notice you have how the heads are interacting with each other. So they intertwine around each other, forming a dimer. You have 2 of these dimers forming a tetramer. Again, head to tail. And then you have the formation of a protofilament where you have this interaction between head to tail again, same idea. So forming a protofilament. Now 8 of these protofilaments can aggregate, forming a large filament. So notice that in contrast to actin, microfilaments, and microtubules, there is no polarity. Now, these intermediate filaments can interact with each other. I already told you that you have type 1 and type 2 keratins. They can assemble together, forming heterodimers. Type 3 proteins can also form filaments containing only a single polypeptide, like bimentin, or you can have 2 proteins forming a heteromer or heteropolymer. Okay? Now, so if we compare intermediate filaments to actins and microtubules, intermediate filaments are more stable, they are more dynamic, they are not regulated by GTP or ATP, okay, like microtubules and actin respectively, but they can be regulated by phosphorylation. So when the heads and tails are phosphorylated, you have the depolymerization or dissociation or disassembly of the intermediate filament structure. Now, something about the intermediate filaments is that they interact not only with each other, but they can also interact with the other structures in cells, the other cytoskeletal components of cells. So they can interact with the actin microfilaments, or they can interact with microtubules as well. And they can also interact with the plasma membrane. Okay? So here's an example of actin structure in muscle cells. Okay, so you have the actin filaments forming an actin fiber, and these structures can be connected to each other via an intermediate filament like desmin, that's in muscles. So they seem to have a function in the contraction of the actin filaments in muscles. Now here's another example where you have a long axon of neurons, and this structure can be stabilized by not only microtubules in green in here, okay, and not only by the actin filaments, as you can see in here, but also you have this interaction between microtubules and actin filaments via an intermediate filament and the different interacting proteins. So you have this cross-linking between microtubules and actin, solidifying and stabilizing the structure of axons of nerve cells. Now, intermediate filaments are also important in cell-cell contact and in cell matrix contact as well. So in terms of cell-cell contact, intermediate filaments participate in the formation of desmosomes. And these desmosomes are made of specific proteins that extend in to out of the cells, and they interact with each other between neighboring cells. And the ends of these proteins, regardless of what the name is, the ends of these proteins can interact with intermediate filaments. So this stabilizes the cell-cell contacts, okay, and you can find it primarily in epithelial cells. These are known as desmosomes, okay? So that strengthens the interaction, the mechanical strength of tissues. Now you also have what is known as hemidesmosomes, whereby the intermediate filaments of cells can interact with matrix proteins outside of cells via integrins, as well as other intermediate filament interacting proteins, such as plaquin or the plaquin family. Now, any defect in intermediate filaments can result in a number of diseases. And it seems that the main function of intermediate filaments really is to strengthen the cytoskeleton of cells and to maintain the integrity of tissues. It doesn't seem to have any role in regulating cell growth and division, because it has been found that a deficiency of intermediate filaments in cells does not affect cell growth and proliferation and division. Rather, what happens in mice that have intermediate filaments knocked down, meaning that mice that do not express intermediate filaments, like keratins, whatever, that they grow fine except that they have blisters or like injuries and lesions in their skin. Okay, so that could be caused by mechanical trauma. Okay, and you can see this happening actually in a condition known as human epidermolysis bullosa simplex, which is caused by mutation in the keratin gene, so that the protein really is defective and not very rigid. So you can see the distribution of different blisters in the limbs of these individuals. Another disease caused by defect in neurofilaments is the amyotrophic lateral sclerosis, also known as Lou Gehrig's disease. And one of the most famous individuals affected with this condition is Stephen Hawking, who wrote the very influential book known as or titled A Brief History of Time. Now, by the way, Stephen Hawking was thought to be dead at an early age when this disease was identified, and he lived for I don't know how many years, but for a long time, and he was quite genius. So again, this summarizes the two lectures that we just talked about. We have microtubules, microfilaments, and intermediate filaments. You don't have to memorize this in here. Just look at the differences. Focus on the characteristics that we just talked about in this lecture. Assalamu alaikum.